Welcome back to another video tutorial on DNA structure and function, which is called as blueprint of life. Okay, now let's get started. So what is DNA? So the full form of DNA is deoxyribonucleic acid, and it is the genetic material in all the living organisms. Genetic material in the sense, this is the material which is obtained from our parents, and it's present in all the living organism like plants, animals, including humans. Okay, and it is the one which contains instructions for growth, development and reproduction. Yes, now when a baby is born, when a newborn when you see a newborn baby, so we need not teach anything to it. Slowly it will start its growth, it will grow, so it will develop, it will start talking, it start walking, right? Nobody is teaching, so everything is happening on its own. And so inside its body also, so the heart is functioning, blood is circulating, all the organs are working. So who is teaching all this to that baby? It is all happening because of the DNA. So DNA, I can just tell it has a biological instruction manual. So it contains the complete information, complete guide, how the growth has to happen, development has to happen, and how the body has to function. Okay, so everything is present in DNA. Fine, and as I told, all living things have DNA in their cells. Fine, and some viruses carry it with them too. Yes, so but in some viruses, RNA also acts as a genetic material, but most of the viruses do contain DNA. Okay, so now as I told, where do we get DNA from? We get it from our parents. Okay, so we get half of it from your mother, from our mother, and half will be from the father. Okay. Okay, now where is this DNA present? So DNA is present in the nucleus of the cell. Mainly it's found in the nucleus of the cell. So as we all know that our entire body is made up of millions and millions of cells, and all the cells of our body will contain DNA. Okay, so where is it located exactly? It's located in the nucleus of the cell. So if this is, as you can see the picture over here, so this is the cell, and inside the cell you can find this nucleus, and inside nucleus you can find the DNA present. Okay, so how is that in the sense? See, inside the nucleus DNA is organized into chromosomes. So it's not directly lying, so but it is organized in a particular form which is called as chromosomes. Okay, so when I zoom into chromosome here, see, this is how the chromosome looks. Okay, when you unwrap the chromosomes, so this is tightly bound, DNA will be completely wrapped and it is tightly bound to make the chromosomes. When you unwrap the chromosomes, so this is how the DNA comes out of it. Okay, so this is your DNA which you can see here. And apart from that, a small amounts of DNA you can also find it in the mitochondria. So mitochondria, as we know, it's the powerhouse of the cell. So inside mitochondria also you can find DNA, and in plant cell you can find DNA in the chloroplasts. So as you can see in the picture over here, this is a chloroplast, and chloroplast also contains DNA, and mitochondria, as you can see, mitochondria also contains DNA. Okay, so whereas in the cells where there is no nucleus, when you take prokaryotic cell like bacterial cell, so there is no nucleus present in them. If there is no nucleus present, then the DNA will be present in the cytoplasm of the cell. It will be present in the cytoplasm itself. Okay, then. Okay, so now coming to the discovery of DNA. So how does the DNA look? What is the shape of DNA? So all this was given by James Watson and Francis Crick in the year 1953. So now how did all this begin it was started with Rosalind Franklin. Okay, she was actually a X-ray crystallographer. So that means she used to study the crystals and fibers using a technique which is called as X-ray diffraction. Okay, she also started studying DNA by using this X-ray diffraction technique, and that's when she will find in one of her photos, she will find a beautiful pattern of DNA. So as you can see in the picture here, beautiful pattern of DNA. So which strongly indicated that DNA had a helical structure. So using this X-ray crystallography images, so Rosalind, this James Watson and Francis Crick, so they'll make a detailed analysis and they'll give the shape and the structure of DNA. Okay, so now we'll study in detail about the Watson and Crick's model of DNA. So yes, the first point, it is a double helical structure or double helix structure, so which contains two polynucleotide strands and which is coiled around each other, forming a right-handed helix. So yes, as you can see in the picture over here, so it is a double helical structure. There is two strands, you can see one in blue color, this is one strand, and the second strand, so which is in green color, this is the second strand. So this is one strand and this is the second strand. So two polynucleotide strands are present here and they are wound around each other, or they're coiled around each other in the right-handed helix. As you can see, they're been rotated or coiled in the right hand orientation, in the right-handed helix. Okay, and the second point is they're anti-parallel. So one strand runs from 5 prime to 3 prime direction and the other strand runs from 3 prime to 5 prime, and that's why we call it as anti-parallel arrangement. That means you can see now this green strand, so it starts from 5 prime and eventually it ends at 3 prime. And so when you take this blue strand, it is the opposite of that. Or in simple words, if I want to tell, so if one strand is starting from head and it is ending with tail, so the other strand so will be ulta. So the tail will be in the top and the head will be down. So this type of arrangement we call it as anti-parallel arrangement. Okay, and then sugar phosphate backbone. Now when you take the outer part of the DNA, now I told, so the outer part of the DNA, these two strands are coiled, they are wound around each other, and these two strands are made up of alternating sugar and phosphate groups. So as you can see in this picture over here, so this is first strand, this is another strand. So you can see there is sugar, phosphate, sugar, phosphate, sugar, phosphate. So like this, the strands, the outer strands are made up of alternating sugar and phosphate groups. And this is the one which gives strength and stability to DNA. Okay, so now coming to the fourth point, see nitrogen bases inside. Bases face inward like steps of a ladder, and 4 bases are present: adenine, thymine, cytosine, guanine. Yes. Now when we come to the inside phase of the DNA, when I take inside DNA, so as you can see, so there are nitrogen bases. See this A, T, so all these are nitrogen bases and they are arranged in the form of a ladder. So you can see, how does this look? This looks like the steps in the ladder. So that's how the nitrogenous bases are arranged. So and there are 4 bases which you can find here. So first is adenine, so which is A, second is thymine, which is T, third is cytosine, which is C, and fourth is guanine, which is G. Okay, and then complementary base pairing, adenine pairs with thymine with 2 hydrogen bonds and cytosine pairs with guanine with 3 hydrogen bonds, and this is always fixed. Okay, always when there is adenine this side, thymine will pair up that side, and that too with 2 hydrogen bonds. Here as you can see in between, how many bonds are there here? There is only 2 bonds. So always adenine pairs with thymine with 2 hydrogen bonds, and if it is guanine on this side, always cytosine will be on the other side. So that means always guanine pairs with cytosine with 3 hydrogen bonds. So you can see the entire thing. See, guanine, cytosine, 3 hydrogen bonds, thymine, adenine, 2 hydrogen bonds. It doesn't matter, adenine only has to be this side. So it can be that side or this side, but always adenine pairs with thymine and guanine pairs with cytosine. Here with 2 hydrogen bonds and there with 3 hydrogen bonds. So this is what continues throughout the DNA. Okay, and then so now coming to the helical dimensions, the distance between base pairs is 0.4 nanometers. So that means now see, so here there is one base pair and here there is one base pair. The distance between both the base pairs here is 0.34 nanometers, or I can tell it is 3.4 angstrom. So 1 angstrom is equal to 0.1 nanometers. Okay, so this is the distance. When you take the distance between any base pairs here, any two base pairs here, so it is always fixed, it is 0.34 nanometers. Okay, and so when you take one complete turn, it is 3.4 nanometers. So when you take this one complete turn, so when one polynucleotide strand, when you see when it takes one complete turn, so it would be 34 Armstrong, or I can tell 3.4 nanometers, and one complete turn will have 10 base pairs. It will have 10 base pairs, and so the diameter of the DNA will be 20 Armstrong or 2 nanometers. Okay, and we can also divide them into major and minor grooves, as you can see here. So this is the minor groove, the small space over here is the minor groove, and this is the major groove, as you can see. So this large curve over here is the major groove. Okay, so again, so this smaller spacing over here will be minor groove, and this will be major groove. So we can divide it into major as well as minor grooves.